Great to be here today to chat about some of the challenges with rare disease drug development. What I'd like to do is to take a minute and ask Emily to introduce Barth Syndrome Foundation.
So thank you for, I'm sure my mic's on. Can you hear me?
Yep.
Okay, good. Thank you for having me here today. Barth Syndrome Foundation was founded in 2000 by a group of parents who were advocating for a better life for their children suffering from Barth syndrome. Our goal is to advance treatments and ultimately find a cure for our ultra-rare, devastating disorder. We primarily focus on R&D and early stage development, and we also partner with our colleagues in industry as well as academia to advocate for therapies and identify opportunities to progress programs forward. We are very small. We have funded around $6 million since our inception. And for those in ultra-rare, I know there's many different organizations here who represent different disease groups. That either sounds really big or really small, depending on where you fall in the continuum. And we have, on any given day, roughly 5 people working. So it's quite an endeavor to try to advance these different programs forward in this very limited capital environment.
Thanks, Emily. Whoa, that's also loud. So I'm with Stealth BioTherapeutics. It's so loud. We were founded in 2006 to develop therapies to address mitochondrial diseases. Mitochondrial diseases have been a very challenging therapeutic target. These can be rare genetic diseases. They can be small or large diseases that you might have heard of, like Duchenne or Friedreich's ataxia have a mitochondrial component. They can also be diseases of aging, like heart failure, which has a mitochondrial component. So many opportunities to develop, but no therapies have yet been approved specifically targeting mitochondria. So it has been a challenging therapeutic area. We kicked off our Barth syndrome program at the request of the Barth Syndrome Foundation and Johns Hopkins in around 2014. Our thinking at the time, we had other programs in development, was, we would do a trial. If we saw a signal, it could be a good bolt-on indication to some of our larger disease indications. And if we didn't see a signal, we would just move forward. The challenge is we maybe saw a signal, right? And in an ultra-rare disease, that left us with a dilemma because it's a challenging investment thesis to continue doing additional trials and investing additional money in an ultra-rare disease. And yet our mission is to develop therapies for patients. And we were hearing from the patients, from the Barth Syndrome Foundation, and from the investigator in her team that patients were seeing milestone achievements on open label extension with our drug. So those are some of the challenges we'd like to talk about with you guys today. When you're in that situation with an ultra-rare disease, hit between what is your investment thesis and what are patients experiencing, where do you go, and what kinds of challenges do you face?
There's no therapies at all for Barth syndrome. Our son's living with it daily. We just need something.
Hey guys, I'm May. I'm here to tell you about this little thingy I have called Barth syndrome.
Hi, I'm Erin and I'm Abe's mom.
Hi, I'm Brian and I'm Abe's dad. Abe is living with a rare condition called Barth syndrome.
From the very beginning, since Abe was born, we had instinctually thought that something was not right. He was in severe heart failure.
It became clear to us that it might be Barth syndrome, so we had the infectious disease doctor help us round up the other doctors and ask them if that's what it was, and we finally came to a consensus that that's what it was.
It kind of set in that we need to be asking about, how do we fix this? And there isn't a fix.
It does affect my energy, muscles, and heart, meaning that I can't do some things as well as you guys can.
So again, I think we've heard today that patients are the best experts, right, on their own rare diseases. And so thank you to Abe and his family and the Barth Syndrome Foundation for allowing them to introduce the disease. I'll ask Emily to talk a little bit more about just how rare this disease is.
Yes, so again, my own gratitude to the Drake family for their help with presenting that. The Barth is ultra rare in the sense that we know about 300 people that are affected with the disorder, and that's across around 3 dozen different countries around the world. Obviously, there's a concentration in the United States as well as Western Europe, and that's driven a lot in part by the diagnosis capabilities that exist in these areas, as well as the medical infrastructure, because it is life-threatening and obviously needs significant experts that can help with the multi-system disorder in its care. So a little bit about Barth syndrome. Unfortunately, 85% of the deaths that occur in our population happen by the age of 5. So if you think of date of diagnosis around 5 months to 15 months, it gives very little time to test interventions in this population. One of the cardinal symptoms is cardiomyopathy. Over 90% of our population suffers from a cardiac disorder. But that's part of a constellation of other symptoms, which include chronic and cyclical neutropenia, musculoskeletal myopathies, debilitating fatigue, and pain, not to mention aberrant growth patterns. And I'm not going to read this for you. This was aggregated during our patient-focused drug development meeting back in 2018, but I do want to call out, it's not just threatening of life and the mortality factor, but there's also the morbidity that weighs into this. And one parent told investigators that my son is not strong enough to open up a bag of chips. He can't even pop a cap on a ketchup bottle. So that just shows what level of impairment they have in integrating with society and experiencing a childhood that we would want for all children.
So we've heard a lot about partnering with patients, right? And certainly in an ultra-rare disease setting, you can't actually find patients unless you partner with patient advocacy. I just want to give a brief shout out before Emily takes you through some of the work that they've done at Barth Syndrome Foundation. Very small, very mighty. I would say that Barth Syndrome Foundation, and we work in a number of rare diseases, has almost followed sort of a textbook, playbook of what you should do. The types of advocacy, the types of natural history studies, the types of driving genetic diagnoses, the types of improving medical education of doctors to drive awareness of this disease, as well as their work partnering with industry. So for such a small organization, you guys have done an amazing amount of work.
Thank you.
Yeah.
I appreciate that. We definitely try to punch above our weight, so to speak. One of the fortunate sides of Barth syndrome, I'll say, is that we actually know what causes it, and that is a mutation or a deletion of the tafazin gene. We've known that since '96. So that's one plus for us. Unfortunately, though, and I'm not sure what the right statistic is. Every time I've heard a presentation, there's a little variation in the numbers today, but we're in that above 90% in way of ultra-rare diseases, or rare diseases that does not have an approved indication. And that's where this partnership is really important for our community to have that hope of ultimately realizing that dream one day. What we have been focused on is promoting awareness and bringing professionals into our realm, so to speak, around the globe so that they can treat as well as also endeavor for research activities.
And I was going to say, on the partnering with industry front, it's not just been industry, but you've had an extraordinary number of interactions with the FDA.
Yes, so this, I don't want to bore you, but I actually want to prove a point with this. We have done a lot of work engaging FDA over the last 4.5 years, give or take. And in part, that we want to plan for success, right? If you're successful in developing a drug, we want to be able to be a good partner in that process of engaging the FDA, educating them on the complexities of what Barth syndrome entails, and also which informs our clinical trial designs. So a couple of things we've done. In 2018, we had an externally led patient-focused drug development meeting. It was no small feat to get the FDA to come to Florida, where we were having an international conference for that meeting. They did, and props to Janet Woodcock for making that happen. And 25% of our global population was there along with their families. So great representation, engagement with FDA to hear directly some of those quotes that you saw earlier. We also then in 2019 had a listening session with FDA. We hand-delivered our Voice of the Patient summary report, which we had generated from the patient-focused drug development meeting. That was instrumental. We've been at the table with sponsor-led meetings with FDA year over year.
We've had a lot of them.
We've had a lot of them, a lot of emails, a lot of meetings.
Yeah.
And even more recently, we had another listening session with the FDA in which our community actually talked to them about tolerance for uncertainty of clinical benefit. We know that there's going to be non-responders. We know that there's risk, especially when you can only have single-digit clinical trials or maybe 12 people in clinical trial. Where was their tolerance on that? So we had direct conversation with them. Even more recently, then we also did a petition of over 4,200 people. So think about it, 300 people in the world diagnosed with this, 4,200 people, because they touch Barth families. They have an understanding of what this means to them, signed a petition pleading with FDA to provide a full review of an NDA for this one drug, elameprotide. We even organized, and I'm sorry, I told you it was a litany of different things we've been doing. We even engaged our healthcare providers, our experts, in appealing to FDA with a letter, which they co-signed. We most recently had a half-day workshop with them, in which we had several key opinion leaders there to educate about prevalence, incidence, burden of disease, all of these different factors that weigh into clinical trials and a regulatory decision-making path, we would hope. And despite that, I would say we are still navigating a very murky labyrinth of what constitutes a regulatory path for ultra-rare.
Yeah. So again, I think that for us, when we talk about rare disease drug development, the challenges of it, what strikes me is that if you're in an ultra-rare disease setting as a drug developer, it's a journey of discovery. We've talked about paucity of, validated animal models, either from an FDA perspective or frankly ones that recapitulate all the aspects of the disease. So the best you can kind of do in animal models in some of these ultra-rare diseases is kind of a gut check that you're not going to run into unexpected safety issues. It's going to be very hard to use them to predict clinical trial designs. We've certainly also looked at cell models like iPSC-derived patient cells, but again, not as predictive in terms of trial design. And so I think that you're going to be learning something in the clinic in patients is what you're going to do. And if it's not perfect, if it doesn't hit a P of less than 0.05, and again, 12-patient clinical trial is about 10% of the US Barth patient population. If you think about doing that trial in heart failure, you're looking at 600,000 patients, right? So small trials because of the small number of patients. And if you're not hitting stat sig, what do you do with that? What if you learn things during the trial? In our case, we designed a 3-month trial. It wasn't long enough. We started seeing meaningful improvements in open label extension. How do you get that data to meet a substantial evidence of effectiveness definition? What creative ways can you use to do that? So these were some of the challenges for us in terms of developing a drug for an ultra-rare disease. No clear regulatory pathway, no animal models, investment thesis, literally investors have said, why are you bothering? You're never gonna really recoup your investment, barring some incentives like priority review voucher. We do have rare pediatric designation, but there's so much uncertainty around it that sort of return on investment starts to dwindle over time. Emily, I know you have thoughts on this as well.
Yeah. I definitely do. We do have animal models, though, so Barth syndrome is monogenic. It's an X-linked disorder. And even though you're dealing with one gene, you have different mutations of that gene or a full deletion. So the genotype leads to different severity, and there's heterogeneity in its manifestations. And that's where you can't begin to create an animal model that's representative of the population. And so it doesn't translate per se into efficacy that we would observe then in a human context.
Exactly. And we've certainly heard, so our journey through the FDA, we started in the Division of Neurology Products. We were moved when we got our phase 2 data over into inborn errors of metabolism, which then reorganized into rare disease and medical genetics. And we're now in cardiology. So over 4 years, we've been in 4 different review divisions with this one set of data. The move to cardiology was initiated by us because we saw an unexpected cardiac signal. So ultra-rare disease, most patients die by the age of 5. A lot of focus was on sort of the pediatric phenotype, which is dilated cardiomyopathy. In mitochondrial disease, it's very common to see phenotypic evolution of cardiomyopathy. And so really in our trial, for the first time, we discovered that for older patients, while their ejection fractions, like the amount of the percentage of blood coming in that's then pumped out is pretty normal, there's not much blood coming into the heart. So the filling is very low. So for our patients, most of them were essentially in congestive heart failure. These were 12 to 31-year-old patients, very close to cardiogenic shock. And so that's a learning from this program that we wouldn't have expected. We've also heard FDA say to us, and Ultragenyx, I think, or others have maybe explored this, think about individualized endpoints for each patient. Well, that's kind of easier said than done if you're trying to pre-specify these endpoints. Another learning from our trial was that some of these patients suffer from nighttime enuresis or bedwetting, which resolved on therapy. But that wasn't necessarily well articulated in the patient community going into the study. So again, I think that concept of a journey of discovery, and then in a setting where you can't really learn from a phase 2 to inform a phase 3, because it's really hard to find the patients or the money actually to mount a second development effort. I'm going to touch a little bit briefly on the data that we had just to give you some background. So again, we saw improvement over 4 years in open label of over 40% in left ventricular stroke volume. The Barth Syndrome Foundation textbook case did a lot of work to build natural history databases, which we were then able to mine and construct natural history controls for our data. So we would expect stroke volume to continue to decline in these patients over time. We had seen significant improvements, like milestone type improvements, again, open label data in patients' ability to walk. So more than 25% improvement in 6-minute walk test, improvements in strength, improvements in balance. Following FDA guidance, we constructed a prognostically matched natural history control study where we essentially matched patients to natural history controls, and we see that these changes wouldn't be expected in the natural course of the disease. The FDA challenge with this, despite their guidance, is that you can't construct a natural history control that's going to address a placebo response on effort-dependent endpoints. So the concept of real-world data and natural histories is great in theory, but it just doesn't really check all of the rigorous statistical boxes that substantial evidence of effectiveness suggests that the FDA should check. And so FDA flexibility in rare diseases, again, that construct of flexibility is very subjective. And so you can have different divisions or different individuals at different divisions interpret that differently. And it's one of the big challenges, I would say, as a developer in rare disease drug development, that, again, you can follow guidance, and in some cases, it may be sort of, good enough, and in other cases, it isn't. So just in terms of things you can do in these ultra-rare diseases, I think we try to be creative, we try to partner with the FDA, but at the end of the day, the standards just may not always be fit for purpose when the disease is ultra-rare.
I mean, I'll just add that the example of which you provided, the bedwetting, I mean, how many children would offer that visiting a doctor? Not a whole lot. And it's also embarrassing for a 13-year-old boy to confess that, especially if his mother's in the room. And it's just not an experience they want to go through. And yet, you couple that with these statistics, and it's evident to me that there's something that is materially different in their quality of life. Something is happening that's giving them more control. And that is where I wish so much that the FDA would be more open to, wow, this actually means something. How can we incorporate this outside of the box that we are used to operating within, especially with larger indications, to learn from this and make a let the gut have a little bit, no pun intended, a little bit of play here in figuring out, is there something that we should pursue? So that's one area. And I do want to mention one other thing. We've had the great fortune of having 2 clinical trials. One is a repurposed drug, which we did out of the UK in Bristol. It's a drug called bezafibrate, and then also this new investigational drug. And the profile for the people that we recruited was almost identical.
Yeah,
almost
very
identical.
similar. Yeah.
And the bezafibrate is a pill. You can take a pill, and elamiprotide is an injection. Well, nobody, not only did it not meet its endpoints, bezafibrate didn't, but nobody's felt any benefit. They didn't want to continue on drug. Lower barrier to adoption, and yet here's an injection they have to go through, children, teens, young adults, and they are begging to stay on it. So I do think when we say the patient voice matters, well, how does it matter? Here you hear people pleading to stay on drug because they perceive a benefit, and there's these other endpoints which aren't formalized that have meaningful, it's meaningful to them, their quality of life. So I guess my point would be to advocate for an openness of new data points that emerge over the course of a trial.
Yeah, so I mean, I think that it's interesting, right? Because substantial evidence of effectiveness, usually you want 2 trials. I think FDA has been really flexible, not adhering to that in the rare disease setting. But pre-specified endpoints, again, you can have surprises. And if you're seeing stat-sig results, can there be flexibility there? The time course to seeing improvements, biological plausibility, natural history controls, all of these tools are out there, but it does become just an exercise of individual discretion. We've heard from senior people at the FDA that they dislike the accelerated approval pathway because sponsors, have not necessarily executed on their commitments, and it's very challenging for the FDA to pull drugs once approved, and we respect that. We've had the FDA say that they think that the ethics of doing trials in ultra-rare diseases like Barth syndrome are similarly challenging as in cases where the animal rule is applied for drugs to treat, biological threats like anthrax, because you just can't necessarily generate the data and the robust data that is necessary. So, I just think that there's barriers across the board in the ultra-rare context, but I think that better consistency of interpretation, better predictability from an investor perspective is somewhat essential to enable further development in this space. It's challenging, and I think again every time you turn around there's a new discovery, whether it's on the regulatory front or on the clinical front. So we'd love to sort of see if there's any questions from the audience or from any of you folks about what we've experienced or other suggestions, thoughts.
And things that we can do, this is, I know at the 3:30 hour, whatever time it is right now, to ask for an interactive is somewhat challenging, but I'm going to challenge you because we have followed a lot of the advice we received throughout today's meeting. Engage patient groups early on, have them be at the table, have them help design trials that are viable, and educate the FDA. Now we've had to do that with several divisions.
4.
It's been an ongoing process, right? But I do think that there's also this issue around grandfathering in certain recommendations because there's clearly turnover. There's inconsistency in staffing, which is a normal pattern in any company, any organization, agency, and that's posed a real challenge for us because despite the educating part of that and bringing the patients to them to hear them, there's also the challenges of maintaining that line of thought through different...
You were asked recently, is it really that rare, weren't you?
We just had to have a workshop for the first half hour to convince them that it's ultra rare. And I basically said, we can't progress to clinical trial design unless we, A, agree that this is an ultra rare indication, and B, that there's a severe unmet need. 2 qualifiers, by the way, for that would point towards accelerated approval being a possibility. And if we can't agree on some of the most simple parameters in which we have to operate, it's futile to discuss clinical trials for our population. The idea of just going to find more patients, which we've heard, quote unquote, time and time again, is advice that I've grown weary on hearing.
Got a question?
Yeah, Samantha from Alexion. I want to bring back a question from earlier in the day. I love the story that you described and all of the work and the advice that you took. One thing I'm wondering about, though, is at what point do we start talking to the payers, right? Because at the beginning of a clinical trial, that's obviously not top of mind. It's how to enroll patients, especially when there's only 300 of them.
Yeah,
right?
so we've had conversations with payers about this disease. Frankly, again, back at the beginning of our development, we were working in a larger indication, which we're still in, which was mitochondrial myopathy. We had a failure there. We're back in phase 3 in a more targeted patient population. Much bigger indication, we can do another trial. But in the context of those discussions, we did talk with payers. We were focusing, again, in the beginning of our development efforts on more like exercise tolerance endpoints, because that's what was known to be the issue in older boys with Barth syndrome. When they understood that cardiac was a big issue, 90% of the patients, both younger and older, die of cardiac reasons, they asked us, do you have any cardiac endpoints? And we were starting to see emerging data. And they said, well, then that's an obvious, sort of fit. So I think we did have early conversations, but obviously, approval comes first, right? Other questions for folks? I mean, when we talk real-world evidence, like what are people's thoughts on how you would incorporate that? And is that more to inform trial design, or do we really think that things like real-world evidence or natural history can serve as controls in ultra-rare diseases?
I'll mention that we have 2 multidisciplinary clinics, as you saw in our chart, and depending on how large of a radius you draw, you can draw that around Johns Hopkins as well as in the UK where we have Bristol. And that has helped to aggregate longitudinal data. And that's very important for a large subset of our population, which introduces a possibility of real-world data.
Yes, for sure. And definitely natural history controls.
And we saw some statistics earlier, I believe you showed, regarding some of those comparisons. And it's not trivial, and yet I believe, has the FDA even looked at it yet? I don't know.
Their concern with the natural history control study we did, which was a phase 3, was that effort-dependent endpoints, so it met its primary and secondary endpoints, but that you can't really control for placebo effect. So on effort-dependent endpoints, that was challenging. I think we have yet to get them to focus on the natural history controls for cardiac. You have a question?
I do. I'm Monica Weldon with the SYNGAP 1 Foundation. So right now, our organization is focused on collecting clinical data because of, obviously, with ultra-rare, because our SYNGAP 1 is an ultra-rare disorder. I'm just curious to find out, have you ever done, or have y'all ever participated in like cross-condition studies simultaneously? Because currently right now, our organization had funded a GATE study with Baylor College of Medicine, a small study between SHANK3, which is Phelan-McDermott syndrome, and SYNGAP 1. And of course, they're still working on the results. Now I was a pretty proud mom at this time, but I have twins, so one is neurotypical, and of course my son has SYNGAP 1. we came up with their study just as like a test run, so they're going to continue on to recruit more. But they found a potential gate endpoint with doing the twins. So that's one of my questions, to find, kind of like killing two birds with one stone, right? So finding a condition that is similar to yours, running these clinical studies to try to find similar endpoints, because possibly they may have a repurposed drug for both.
Yeah, there's not any therapies approved for mitochondrial diseases. So I would say we came to Barth from the construct of it was called mitochondrial myopathy, all different genetic mutations that caused exercise intolerance, can also manifest as cardiomyopathy and neuro issues. So we had done a basket trial in a larger indication, and we actually, that trial failed because of the variability between different genetic mutations, frankly. So we saw a subgroup of responders, but like the heterogeneity of the disease presentation, particularly with different genetic mutations, was sort of fatal to the effort. So there can be benefits, like certainly for us, it was a good approach to kind of a blanket approach to this potential problem, but it kind of was overcome by the heterogeneity.
And probably the different function and mechanisms of the gene as well, or the.
Yeah.
I'll say that we've definitely considered that approach. However, I think Reenie touches on something that's really important, is even within, when you've done the genetic panel, you understand where there's an anomaly, even that's just one gene, and still we have such tremendous heterogeneity in there, such that the mouse models are not predictive of the clinical benefit. And even in a group of, give any number, I mean, I'll say 20 for right now, you're liable to have only 60% responders. I mean, I'm just throwing numbers out there.
For any drug.
For any drug, for any drug.
For any drug.
And so that was an approach we had to try to create order of magnitude, so to speak, and alliances of like, we have similar clinical manifestations. Is there something that we could do that could address these underlying causes? And I think earlier today we were talking about specificity versus, I guess for ultra rare or for rare, versus just a general symptomology. And what is the right approach? I'm not ready to write off the symptomology piece of this. I'm just not. But I will say for clinical trials, I would be reluctant, I'd be hesitant at present because of this question of the variability.
And I think you get a progression over time with these diseases too. So even talking to FDA, they've asked us, well, you could enroll infants. Well, number one, you kind of got to catch them when they're born. So how do you set up a trial to do that? But number two, you can't look at the same endpoints. The disease presentation is different at different ages. So again, it all kind of adds to the complexity, I think, in these very small patient populations. Anyone else? Ultragenics, I know, looked at multi-domain responder indexes, right, which we did in our phase 3 natural history study. But our experience with FDA was that they were questioning those. While encouraging novel endpoints like that, we also had a lot of skepticism around them and need to validate them, which adds time and development challenges. So, yeah.
I'll add that it'll be a tragedy for this population if we can't identify ways to advance a product forward that has demonstrated already such tremendous impact for, again, a handful of patients. If we have to have a multi-site trial, if that's been something they've actually told us, maybe consider in the future as a modus operandi, is that we have to have a site here in the US, in the UK. I know there's investors in this room. I know there's pharma in this room. Can you imagine that being the prospect going forward of how we have to design trials, given the complexity? I don't even think about GDPR right now, and other EU regulations. Like, it just blows my mind, the added complexity. It's hard enough in this country, in a single site, and I do appreciate why multiple sites are part of recommendations. But for ultra rare, when you're talking about
20 patients.
Well, you
Give
just
me a
can't
break.
make, you can't make that money back. I mean, unfortunately, that's what it comes down to, is you can't really make that money back. So it's definitely challenging. I think it'll be a challenge. I mean, it's interesting as we think about more N of 1 diseases, right, and individualized medicine. I don't think this is a challenge that's limited to Barth syndrome or some other similarly rare diseases. I think it's going to be something we're going to have to face, right, more generally in the industry as personalized medicine becomes more normalized and science progresses to enable it. Great. Thanks, guys.
Yeah.